Okay, hello everyone. Welcome to Creative Biolabs Lab webinar series. And I would like to introduce Sarah Vogelan first, who will briefly present our company's service and products.
Thank you, Kathy. Welcome everyone. As a start of this webinar, I will indeed give a brief introduction of our products and services related to our Therapeutics platform. So Creative Biolabs offers several services to assist your projects that are focused on solid tumor targeting, including CAR T development and TCR-T development for solid tumors. So our CAR T services include biomarker identification and scFv generation, CAR design and construction, gene packaging and delivery, and robust in vitro and preclinical in vivo assays. In addition, the TCR-T development services, for that we have neoantigen identification and TCR discovery, TCR affinity optimization, functional characterization, as well as preclinical and clinical translation support. Furthermore, we offer specialized CAR designs for solid tumors, including options to target multiple antigens simultaneously, such as the tandem CARs or multivalent CARs. We can also help with improving CAR T cell infiltration and persistence through chemokine receptor modifications or armored CARs, enhancing targeting specificity and reducing off-target toxicity through logic-gated CARs or on or off-switch CARs or switchable CAR T cells, and addressing immunosuppression in the tumor environment through disrupting inhibitory pathways and overcoming hypoxia. We also offer specialized TCR-T design strategies for solid tumors, including targeting a broader range of antigens through intercellular and cell surface antigens, neoantigens, and tumor-specific antigens, and overcoming the immunosuppressive tumor microenvironment through engineering of cytokines or targeting immunosuppressive factors. Furthermore, we offer featured products for solid tumor targeting, both for CAR as well as TCR-Ts. For CAR products, we'd like to highlight our CAR vector products, as well as CAR cells, CAR jerket cell products, and CAR MA vector cell products, and also the immune cell products, CAR animal cells, and stable cell lines that we can offer. And for TCR-T products, we have also the TCR vector products, TCR-like CARs, TCR-like antibodies, antibody TCR vector products, TCR jerket cell products, and DNA-encoded monoclonal antibodies. And lastly, I would like to highlight our featured platforms for solid tumor targeting. Again, both for CAR and TCR-T, we have featured platforms such as the Therapeutics CAR technology, the surf array cell microarray technology, and the mRNA-based CAR cell platform. We also offer an animal model platform for CAR T therapeutics and mouse CAR T cells, and CRISPR edited CAR cell technology. And for the TCR-T platform, I'd like to highlight that we have TCR sequencing service, as well as Therapeutics TCR technology, CAR TCR bispecific T cell engineering platform, and T-seeking platform for TCR and ligand discovery solution. And with that, I would like to hand the floor back to Kathy for introduction of our esteemed speaker of today.
Thanks, Sarah. And now let me introduce our speaker for today, Dr. Mark Leick. He's an assistant professor at the Harvard Medical School, assistant in medicine at the MGH in the cellular immunotherapy program, and a principal investigator in the Crest Family Center for Cancer Research. And he splits his time caring for CAR T cell patients on the MG CAR T cell service and in the lab developing novel CAR T cells for cancer and understand the mechanisms of resistance and toxicity to existing CAR T cell therapies. And before Dr. Leick begins his presentation, I want to quickly remind our audience that we will have a Q&A session at the end of the presentation. So if you have any questions or comments during the presentation, you can just type your questions into the Q&A panel, and Dr. Leick will get to them after his talk is finished. So now let us welcome Dr. Mark Leick, and thank you for presenting your important findings to us today. And you may begin when you're ready.
Great, thank you so much for that introduction. Let me just pull up my slides here. There we go. We all set? Everyone can see?
Yeah, looks great.
Great. Thank you. So this is part of a project that we've been working on for a while that consists of multiple overlapping projects that have started in hematologic malignancies and now spread to solid tumor malignancies. So I'll be going through it. These are my disclosures, and so this talk is sponsored by Creative Biolabs. And so I'd like to start off by talking about all of the FDA-approved CAR T-cell therapies that we have in hematologic malignancies. And so I think sometimes people who are primarily lab-based may, or not even connected to CAR T-cell, may think of CAR T-cells as an abstraction. It's just sort of a research tool. It cures some mice. And the reality is that these are FDA approvals in a variety of different blood cancers that we use in the clinic all of the time. And so this is the patient that I saw recently. This is, you don't have to be a radiologist to be able to look at this PET scan where all of the glowing areas are tumor. And then 30 days later, after CAR T-cell therapy, essentially all of the tumor is gone, and you're just left with normal PET radiotracer in the kidneys and bladder. So this is an incredibly powerful therapy that people are very excited about for good reason. And it's founded upon the endogenous signaling of T cells. And so I like to start off by showing people how CAR T cells leverage the endogenous T cell receptor. So canonically in immunology, we think of the T cell receptor as requiring three activation steps. So the first is the TCR engaging with peptide in the context of MHC. So that's signal one. And then signal two is co-stimulation. Shown here is 4-1BB, but there's a variety of different other co-stimulators, the most common of which, besides 4-1BB, is CD28, which you'll see. And then finally, signal three comes in the form of cytokine support. So IL-3 is shown here. The major innovation with CAR T-cell therapies, so-called second-generation CAR T-cell therapies, was the spatial integration of signals 1 and signals 2. Investigators found from the 80s and 90s that if you didn't have that spatial integration, the CAR T cells, they just didn't last that long. They didn't persist. And the fusion of signals 1 and signal 2 made all the difference. And that's the strategy upon which all FDA-approved CAR T cell therapies are built. And so the way that this happens in the hospital is that we bring the patient to get their T cells collected. They undergo what's called an apheresis. So we take out T cells, we send them for manufacturing. The T cells are then activated and exposed to good manufacturing processes. So clinical grade virus that transduces the CAR construct into the T cells. The T cells are then expanded after some quality control. Then they're administered to the patient after lymphodepleting chemotherapy, which makes room for the CAR T cells. And so in the context of kidney cancer, which is an area that my group has been focused on for a little bit, there's been a few different products that have been tried in patients with kidney cancer targeting CD70. And so one of them is this product CTX130, which is an allogeneic CAR T cell targeting CD70. And so the investigators, at this company undertook a variety of different modifications to make this suitable for allogeneic use in patients. So they deleted the T cell receptor so that there wouldn't be any host versus graft versus host disease. And then they deleted the CD70 locus. They found that T cells, when activated, can express CD70. And so they didn't want any fratricide, so T cells killing other T cells. And then finally, they deleted the beta-2 microglobulin locus. And then they did a clinical trial. And so these were patients with relapsed refractory renal cell carcinoma, where CD70 is abundantly expressed. They did a dose escalation trial. And essentially what they saw is that there was maybe one responder, but largely, responses were mixed and underwhelming, which is consistent with what we see in a lot of solid tumor contexts. And this was the biopsies before and after treatment in terms of CD70 positivity. And so you can see extremely high levels of CD70 expression, essentially 100% in the majority of samples. And at day 42, again, still very high expression of CD70. And so in the field, some people have said, if you make a CAR and against a given target and you biopsy after CAR T cell treatment and you still have abundant expression of that target, you just haven't made an effective CAR. And so we know that allogeneic CAR T cells are limited generally in their persistence because despite these modifications, ultimately there's some immune cell recognition of the CAR that limits its persistence. And so another competing company, Allogene, had also made a CD70 targeted CAR, this Allo 316 product, which got fast track designation. And they performed some similar edits, deleting the TCR as well as CD52, because they used the anti-CD52 antibody alemtuzumab as part of the lymphodepleting protocol. And so this was also a dose escalation study. And again, a couple patients had, even significant reductions in disease, but again, only partial responses. Again, somewhat underwhelming. And so from this baseline, looking at renal cell carcinoma, how can we improve things? And so one thing, as a medical oncologist, that in my training I've seen, we use VEGF or vascular endothelial growth factor blockers in a lot of different cancers. So these are all the FDA approved areas where bevacizumab, which is a monoclonal antibody against VEGF, are used, and then it's used off-label in a lot of other diseases. On its own, it's not perfect. It doesn't lead to cures, but it's a definite adjunct to a number of different cancers. And so one challenge with the blockade of VEGF, and so VEGF is important for maintenance and health of your endothelial system, as well as your kidneys and your cardiovascular system. And so the blockade of that, as you could imagine, could lead to some complications, including hypertension, high blood pressure, heart failure, and other problems. So that comes with some consequence. Separately, on the lymphocyte side, we've known for a little while now. So this is a paper from Stephen Rosenberg at the National Cancer Institute, who's a leader in adoptive cell therapy, that actually, if you give adoptive cell therapy with VEGF blockade, you actually enhance infiltration of those adoptively transferred cells. And so this goes along with what people are saying, that VEGF blockade kind of normalizes the tumor vasculature and enhances infiltration of lymphocytes. And then there's also several lines of evidence that suggest that VEGF actively is harmful to T cells and decreases their viability in a dose-dependent fashion. And so this is a patient where they're able to take T cells from the ascites and show that VEGF was detrimental to their function. This has been also shown in other groups that show, again, a dose-dependent increase in exhaustion markers associated with VEGF administration, as well as double positive. These are in animal models that can be ameliorated with VEGF blockade. And then when you when you administer VEGF, you get high levels of exhaustion. And then in this group, they did a really nice conditional knockout of VEGFR2 just in the T cells, showing that in this colorectal cancer model, they had actually decreased tumor when VEGF signaling was lacking on the T cells. Again, another line of evidence suggesting that VEGF is a negative regulator of T cell function, and blocking its effects can enhance anti-tumor activity. And so we know that VEGF can drive a hostile tumor microenvironment for immune effector cells. And so generally a trigger for VEGF secretion can be hypoxia, and this leads to unfavorable polarization of macrophages, increase in exhaustion markers in your lymphocytes, and increase in T regulatory cells, and overall a net unfavorable tumor microenvironment for T cells. And so that led us to the following hypothesis: is that CAR T cell secretion of a VEGF blocking scFv would avoid the systemic toxicity that I talked about with the VEGF blocking drugs, and then prevents T cell exhaustion, leading to enhanced expansion and cytotoxicity. And so there's another group who had done something related, which is again kind of similar to what Steve Rosenberg's group had done. Is what they combined anti-VEGF antibody in mouse models, in syngeneic mouse models of glioblastoma, with CAR T cells, again showing increased infiltration of the CAR T cells, normalization of the vasculature. In terms of targeted secretion of therapeutics from CAR T cells, this has been done in several ways. One is in this nice paper from Sloan Kettering in 2018, investigators had a CAR T cell that secreted a PD-1 blocking scFv and showed that in a peritoneal model, if you administer CAR T cells by tail vein and then you track the scFv with a bioluminescent tag, you can see on the top on A, you can see that the scFv stays over the peritoneum of the mouse, which is where the tumor is. Conversely, if you give just a generic monoclonal antibody that's conjugated to the same reporter, bioluminescent reporter, you can see that it goes all over the mouse again, which is exactly what we don't want because we know that VEGF and some of these molecules are toxic when administered over the entire body. And so our group over at Mass General has pioneered this also in another way in patients in the context of glioblastoma. And so in this clinical trial, we have a CAR targeting EGFR-V3 on the tumor cells, on the glioblastoma cells. And then they secrete this T cell engaging antibody molecule, which is essentially identical in design to a bispecific T cell engager, or BiTE. And on one side, it binds to wild type EGFR, and on the other side, it binds to the T cells, CD3. And so it's capable of engaging either the CAR T cells or T cells within the tumor microenvironment. And so this CAR T cell product is injected directly into the cerebral spinal fluid through the use of an Ommaya catheter, which is implanted into the brain. And so we can access it subcutaneously, and then we inject the CAR T cells into the brain. And if you look by flow cytometry at the T cells that are bound to this TEAM or BiTE molecule, you can see these are readily detectable in the cerebral spinal fluid for a few weeks. Conversely, if you look in the blood for any of that TEAM molecule, you don't see any. So this, again, provides strong evidence that by having CAR T cells secrete a molecule in a localized fashion, you can compartmentalize that and prevent it from leaking out into other undesirable areas where it might lead to toxicity. And so one other line of evidence that we looked at before we got started was our group had previously done a correlative study looking at patients who'd been treated with diffuse large B-cell lymphoma with the two FDA-approved CAR T-cell products at the time, Axi-cell and Tisa-cell. And we collected samples either prior to CAR T-cell infusion, the infusion product itself at day 0, and then 7 days after CAR T-cell infusion, we took the PBMC, peripheral blood mononuclear cells, and then we flow sorted out the CAR T cells to enrich them and then perform single cell RNA sequencing. And so we were able to take this data and then reanalyze it for expression of different VEGF family members, including the receptors. And what we saw, interestingly, was VEGFR2 is the canonical signaling member, which is implicated generally in endothelial cells, the receptor. But what we saw is that actually VEGFR1 was very high in the infusion product, especially in the non-responders of one of the CAR T cell products, suggesting that there may be some correlation with expression of this receptor and response status. And so we then sought to try to replicate this finding in our own hands. And we generated CAR T cells targeting mesothelin with either 4-1BB or CD28 co-stimulation, just like the two products that we had evaluated with single cell RNA sequencing. And similar to what we had seen, we saw higher levels of VEGFR1 in the stimulated CAR T cells with the 4-1BB co-stimulatory domain. And we saw this across different donors. And so in general, we think of the VEGFR1 is classically thought to be a decoy receptor that sort of modulates VEGFR2 signaling. But there's emerging evidence that suggests that actually VEGFR1 on its own has important access to fundamental pathways that control cell migration and proliferation. And so the second part was what if we're going to use this secreted blockade strategy, what is going to be the single chain variable fragment that we're going to use? And so in using this strategy, we thought it would be prudent to use a single chain variable fragment because it's smaller. And in general, in therapeutics that are used in humans, the goal is to have long-lasting therapeutic potential. And so there's a lot of optimization done by pharma companies to make it so that these molecules persist in the body as long as they can. In our case, we actually wanted the opposite. We wanted a short-lived single chain variable fragment because the CAR T cells could make it continuously. And the faster it's cleared from the bloodstream, the less likely that there would be to have toxicity outside the immediate area where the CAR T cells are. And so this is a nice drug that had been developed by Novartis, brolucizumab, which inhibits all isoforms of VEGF. It's quite small, so it has better penetration. It's humanized. It's very stable and highly soluble compared to existing inhibitors. Currently, it's only approved, FDA approved for ophthalmologic use. Again, probably because as a single agent, again, it doesn't persist very long, and so its anti-cancer uses are minimal at the moment. Though it has been tested in small immunocompromised mouse studies. So this is a study where they tested it in a dose-dependent fashion against a glioma model. And this is the same drug again, brolucizumab is DLX1008, showing that it indeed does have some single agent activity. And so one of the first things that we did is we modified our CAR T cells to now secrete this brolucizumab VEGF blocking molecule, and then we developed a VEGF reporter cell line assay, and so we could take these reporter cell lines, and then when we added different levels of recombinant VEGF, we could determine if the supernatant from our anti-VEGF secreting cells could block this. And so what we saw was a range of blockade of VEGF reporter activity across relevant concentrations of VEGF. We could also do this when we take the supernatant from, for example, a renal cell carcinoma tumor. We could effectively block that substantially. We used a CD19 SAFE as a control, which shouldn't have any activity on the VEGF. And then finally, when we expand CAR T cells, and so we do a production expansion for 14 days, and if we compare CARs that make a VEGF blockade versus those with a control blockade or no secreted factor, we see that the CARs that make an anti-VEGF FCFB, when we do an ELISA, effectively we can't detect any VEGF compared to a media control, suggesting that we're achieving high levels of VEGF blockade. And so we wanted to develop a functional assay to be able to determine if our T cells were making a bona fide functional VEGF blocker. And so we worked with an assay that we had developed on the IncuCyte machine, in which we co-cultured human umbilical vein epithelial cells that were GFP labeled with fibroblasts, primary human fibroblasts. And when you add VEGF, this co-culture actually forms primitive blood vessels that can be detected on the IncuCyte and measured in a quantifiable way. In contrast, if you add something that blocks VEGF, you won't get this vessel formation. And this is what this looks like. And so in the top two rows, we can see at hour 0, I'm showing you the fluorescent channel, the GFP HUVEX. You can see the HUVEX, but no blood vessel formation. And then in the software detected vessel, we're showing you what it looks like when the IncuCyte is trying to interpret this data. And we have the anti-VEGF condition as well as the anti-CD19 control, where we took supernatant from CAR T cells producing each of those and then added them to the culture. And then at hour 70, we can see that in the anti-VEGF condition, we've abrogated essentially all blood vessel formation, which you can see is detected by the IncuCyte. And so this is quantified here with the relevant controls. And so, in thinking about how to develop a comprehensive strategy for targeting renal cell carcinoma, we leaned back on a CAR that I had previously developed for acute myeloid leukemia targeting CD70. And so, lots of people, lots of CAR T cells that are all of the ones that are FDA approved and lots in the clinic rely on single chain variable fragments, often from mice and now increasingly camelids. And one challenge with those is we're learning more and more that actually CAR T cells are limited in terms of their persistence and activity by immune responses, endogenous T and B cell responses, despite lymphodepleting chemotherapy. And so we took a different tact and we used the natural ligand to target CD70. And so the natural ligand for CD70 is CD27. This has the added benefit of the fact that CD70 is a tumor necrosis family member, and so it forms signaling hexamers with CD27, which form these higher order polymers on the surface of the structure of the cell. And we thought by using a natural ligand, we might be able to engage in these higher order signaling clusters. And so one problem that we realized early on is that CD27 actually undergoes proteolytic cleavage by matrix metalloproteinases, which are made by tumor cells. And this leads to essentially decapitation of the CAR if you don't address it. And so we did essentially a screen to identify alternative hinge regions that were not susceptible to cleavage by the tumor, identifying the CD8 hinging from its membrane, which decreased this cleavage event and made a much more robust CAR that had substantially higher proliferative capacity. And so it turns out that CD70 is also, in fact, it's most highly expressed on renal cell carcinoma of all tumors. And so we move forward with this CAR. And the superiority of the ligand-based approach has now been demonstrated over the single-chain variable fragment, has now been demonstrated by several groups. In this case, the investigators were comparing ligand-based CARs to single-chain variable fragment-based CARs for CD70, finding that the ligand-based one had the highest expansion. So again, the dark red mice here, this is CAR T cell expansion, which was much higher in the CD27 based targeting moiety on the far right. And then again, another group showing that the ligand-based version again has superior CAR T cell expansion compared to all the other versions. And so to deal with antigen heterogeneity that we'd identified in acute myeloid leukemia, we took this ligand-based CD70 CAR and then had it secrete a T cell engaging antibody molecule or a BiTE essentially targeting CD33. So we developed this pathway to have the CD70 CAR now secrete these biologics to enhance their activity and modulate the tumor microenvironment. And so, as I mentioned, renal cell carcinoma on the far left, KIRC, in the TCGA has the highest level of CD70 expression relative to all of the tumors, much higher than AML that we've been working on. And if you look at, for example, a representative IHC of CD70, you can see it's extremely robust. And then we can look at CD70 expression in some patient-derived xenografts. And so these are essentially patient samples that underwent bulk RNA expression. We can see that, compared to sort of negative control CD19, which is a B cell marker, we see extremely high levels of CD70, even higher than a canonical RCC marker, which is carbonic anhydrase 9. And so the idea here was to have the CAR T cell, and we know that CAR T cells themselves actually make VEGF, that they would secrete this VEGF blocking scFv, and that would block the VEGF from binding its receptor on the CAR T cell, as well as the endothelium, as well as blocking VEGF produced by the tumor cells as well. And so we started doing some assays where we would take CAR T cells targeting mesothelin, again with the anti-VEGF or the anti-CD19 control, and we activated them with either SKOV3, which is an ovarian cancer tumor cell line, or just plate bound. So this is no tumor, just CD70 adhered to the plate. And we can see higher levels of activation, the CD69 activation marker in the VEGF blockade secreting CAR T cells. We also wanted to look at how the presence of the VEGF blocking scFv might perturbate CAR T cell expression profiles. And so using the nanostring gene expression, this is the targeted gene expression profiling technique, we looked across 4 different T cell donors. Again, the CD70 CAR T cells that secrete the VEGF blocking scFv versus those with the sham CD19 control scFv. In the absence of any tumor cells, so this is just plate-bound CD70, we could see, dramatically different expression programs with higher levels, for example, of type 2 interferon gamma signaling in the CD70 CAR secreting the anti-VEGF scFv. Um, we next moved to actual cytotoxicity assays. So again, on the Inge site, we could do these co-cultures and killing assays where we look to see if the CD70 CAR T cells demonstrated enhanced tumor clearance. So GFPs marking the tumor cells, and we saw that the 70 anti-VEGF had enhanced tumor control. And then we can also look at our mCherry reporter on the T cells, on the CAR T cells, to look at CAR T cell expansion. We saw higher levels of CAR T cell expansion. And we could also do the same thing in ovarian cancer. And we can see when we look at cytokines at the end of the co-culture, we have substantially reduced levels of VEGF, suggesting that we're achieving VEGF blockade, and this correlates with enhanced tumor killing and higher levels of CAR T cell expression. We also wanted to look at long-term proliferation. And so in this assay, we took our mesothelin CAR T cells, either the anti-VEGF secreting or the sham CD19, and we can do recursive stimulation. So every 3 days, these CAR T cells would get against additional K562 mesothelin expressing targets that have been irradiated. And then we would add them to the culture and watch their cumulative CAR T cell expansion. And you can see, again, these meso anti-VEGF CAR T cells had substantially higher proliferation. I mentioned when we were doing those cytotoxicity assays, we could see, again, higher levels of killing against SKOV3 and UpCAR3 ovarian cancer cell lines, higher level of tumor cell killing, as well as higher level of CAR T cell expansion. And then when we look at effector cytokines, we can see that we've completely suppressed VEGF, detectable VEGF in both cultures. And then we also have higher levels of interferon gamma, TNF alpha, granzyme B, and IL-2. Again, all cytokines that are typically associated with highly effective CAR T cells. We also looked at a few other tumor types, and so blockade of VEGF has been demonstrated to show some signal of efficacy in acute myeloid leukemia. And so we tested these CAR T cells against a tumor line, NOMO1, which is a type of acute myeloid leukemia. And again, we could see the tumor in green, substantially higher levels of killing with the meso anti-VEGF CAR T cells, and then again, higher levels of proliferation with those CAR T cells as well. We next wanted to move on and test these in vivo. And so we utilized this is a lung cancer metastatic model. And so A549 tumor cells that had a bioluminescent reporter were injected in the tail vein of these mice. And after 7 days, they would receive CAR T cells, either the mesothelin targeted with the anti-VEGF scFv or the control CD19 scFv. And you could see, again, eradication of tumor in the meso-VEGF with residual tumor in those with the sham antibody. And when we phenotype the CAR T cells from the blood of these mice, we see a trend towards different levels of, for example, TIM3, and then higher level of these effector memories cells in the meso anti-VEGF. So again, suggesting that there are differences in the phenotype that are perpetuated by the presence of this VEGF blocker. When we look into the lungs of these mice, and so we sacrificed these mice earlier, And we can look at where the tumor is. So in this metastatic model, the tumor is kind of all over, and so that's stained by CK7, so-called cannonball lesions all over the tumor. Um, and so it makes it hard to quantify the T cells across the entire slide because the tumor is localized into these all these discrete foci. But if you look within the regions of the tumor, you can see infiltration of CD3, human CD3 cells, in all of these regions of tumor. And so these two mice I'm showing here are the anti-VEGF secreting CAR T cells. And you can also see some decrease in murine CD31. So again, our anti-VEGF blocker, brolucizumab, also has activity against murine VEGF. And so we'd expect there to be vascular changes in the mice as well. And so if you compare these images to the mice that received the CAR T cells with the antibody targeting CD19, when you look into the tumors, into the CK7 positive A549 tumor regions, you actually don't see essentially any T cell infiltration. So a dramatic change between the two groups. Separately, we also developed this orthotopic xenografts model of renal cell carcinoma. And so 7860 tumor cells were injected into the renal capsule in these mice. And then after a few weeks, by tail vein, we injected the CAR T cells, either the CD70 with the anti-VEGF scFv, or the anti-CD19 control. And on top, I'm showing you Vimentin staining, which marks the tumor. And then below that is the human CD3 stain, with untransduced T cells being the control, with T cells, activated T cells, but no CAR in them. And then looking at VEGF staining, which is quantified below using QPath, you can see a paucity of VEGF overlying the T cell infiltrative area, suggesting again that there's this localized blockade of VEGF by the secreted T cells. And so if we look at another area, this time in the 70 anti-VEGF, I'm showing you two areas. One area is where the CAR T cells are, and then another identically sized area where there are no CAR T cells. And you can see a significant difference in the amount of VEGF that is expressed in those two regions, with the one overlying the T cells expressing minimal levels of VEGF, while when it's away from the T cells, you can see abundant VEGF. Again, demonstrating the concept that these CAR T cells are achieving this localized blockade of VEGF. And this is quantified here. Again, showing the 70 anti-VEGF have substantially reduced amount of VEGF positive cells in that region. And so in summary, I'll say that activated CAR T cells upregulate VEGF pathway members. Our CAR VEGF, so the CD70 and the mesothelin CAR T cells, secrete a biologically active and potent VEGF blocking scFv. And then CAR VEGF have enhanced in vitro anti-tumor activity and proliferation. And we've also demonstrated that they have enhanced in vivo potency in immune-compromised mouse models of lung cancer. And so with that, I'm happy to take any questions that the group has, and thanks for your time.
Thank you for that great talk, very interesting. So yeah, everybody feel free to post your questions in the chat. And while we wait, let me just share the screen. Yeah. Yes, so please, everybody, feel free to start typing the questions in the chat. And while our attendees are writing their questions, I will start off with one of my own questions. So you mentioned at the earlier part, in the first part of your talk, where you were treating with CAR in the CSF of the brain, and in the brain basically using a catheter. Could you elaborate a little bit on this experiment? Was this done in mice or in humans? And if so, if it was in humans, how are the patients doing? Were there any side effects? Yeah, could you just elaborate a little bit on that?
Sure. I'll pull up the slide again so people know what
Yeah,
we're talking about.
sure. I can
Um,
stop the share.
so this was a clinical trial in patients at our hospital
Mhm.
at Mass
Okay.
General. Um, nope, I don't know why it doesn't. So these were patients. So this report was the first three patients who were treated at our hospital. And so, as you mentioned, the CAR T cells that secrete this bispecific T cell engager or TEAM were injected via this catheter directly into their brains. Um, and so the first three patients were treated. We haven't published the follow up to them yet, but we did report some responses in those first three patients. We didn't have any substantial toxicity, and so so far in this clinical trial, we haven't needed to use any major drugs to deal with CAR T cell toxicity. We haven't needed high dose corticosteroids. We haven't had to use tocilizumab or any of these other things, and so it's been exceptionally well tolerated. And so, we think this is a really viable strategy in solid tumors and many other tumors.
Okay, that's great to hear. And are you so yeah, you mentioned there's nothing really happening in terms of side effects. And are you not worried either about microglia response, for example, because these will respond when you bring in the catheter?
Yeah, you just mean from the catheter itself, or
Yeah, for example, or even to the treatment.
yeah, we haven't seen it. So I will say that patients get what's become known in the field as TIAN or inflammation associated neurologic toxicity. And so we think this is because the T cells will infiltrate the region of the brain where the tumor is, obviously. And that can cause inflammation. And so therefore patients will develop side effects related to where that region is in the brain. So for example, if someone has a tumor that's overlying the language area of their brain. And maybe at diagnosis, they had some issues with speaking. It could be that during their CAR T cell treatment, you might see some of those language difficulties transiently get worse while the CAR T cells infiltrate that area before getting better. But that's kind of been described with other people who are doing brain tumor related CAR T cells. So what we've seen is consistent with that.
Yeah. Okay, great. Thank you for elaborating on that. And then maybe also, another follow-up, more general, to the overall talk. How do you see the field evolving in the next couple of years?
Uh, you mean solid tumor or just CAR T in general
Yes.
or what?
Yeah, so solid tumor and the use of CAR T in solid tumor.
Okay. Well, I'm gonna answer the question you didn't ask, which I think is a little bit easier. I think there's going to be a lot of CAR T cell use in autoimmune disease. I think this is a huge growing area. There's a lot of movement in vivo CAR T cells that I think are going to be extremely popular. I think there's a lot of people talking about that for solid tumors. I don't think it's the right strategy yet. I think if you can't get an autologous CAR T cell to work for a solid tumor, I think it's a fool's errand to try to think that an in vivo CAR T cell is going to achieve that. Again, because the transduction rates are quite low with those strategies. The immune microenvironment in solid tumors it's a lot more complex. And so I think you're going to be seeing a lot more armored CARs. So, we've had secreted therapeutics, but there's a lot of people doing, secreted cytokines, TGF-beta dominant or negative receptors. I think dual targeting, I think elimination or modification of a lot of negative regulators of T cell function are becoming very popular. So I think some of those different options we'll continue to see.
All right, great. Thank you so much. I think there are no further questions at this time. So I think with that, we will wrap up the webinar. So I'm going to hand the floor back to Kathy for the concluding remarks. But thank you, Mark. That was a great talk.
Yeah, of course. Thank you.
Oh, I see some attendees just raise your hand, please. Okay. Could you please type your question into the Q&A panel, and so we can follow your questions quickly. Yeah.
Sorry, I missed that. Somebody raised a hand. I didn't see the questions in the chat yet. Yeah, but please, indeed, everybody who has any questions, feel free to type them in the chat so we can ask them. Okay, we'll give them maybe one more minute to type the questions. Let's see.
Okay, he just find it now, and could you please type your question now? Yeah, we can
Yes.
hold on just a second. Yes.
Okay,
Yes,
so I'm
Kenneth, if
seeing
yeah,
a
yeah.
question. Where do you see things like Alzheimer's and Parkinson's or Parkinson's? Um, So I think this is challenging. So, a lot of people believe, and I am far from an Alzheimer and Parkinson's disease expert, um, but the general understanding of the pathophysiology of those conditions is it's related to abnormal protein buildup in the cells, and that leads to neurons essentially dying. And so, in the general sense, I don't think T cells will necessarily play a role. Um, uh, we think that T cells in terms of the autoimmune space are doing well for diseases where there's a T cell or a B cell autoimmune attack on a specific tissue. But sort of an indirect way of answering your question is there are groups working on CAR macrophages. So macrophages have the ability to engulf proteins and other things within our bodies. And so people have put CARs into those. And so if you could redirect those cells to engulf these bad sort of tangles and plaques in these diseases, then perhaps you could develop a therapeutic benefit. I think that's pretty far from the clinic, but that's one way I could see adoptive cell therapy helping out in those diseases.
All right, great. Thank you for answering that.
You're welcome.
Okay.
Okay,
All right. Yes,
I think
go
we're
ahead.
going to finish. Yes.
Yes.
Yes. Thank you, everyone. Please know that Creative Biolabs regularly invites estimate experts in life science and biotechnology to share the latest findings in our webinar series. So we encourage you to visit our website and subscribe to our social media channel so you won't miss any upcoming events. So that's all today. Thank you, everyone, for joining us, and we will see you next time.